questions yesterday. Who asked that question about the sewage, biofilm function in sewage? One of you asked that question. How biofilm help improving sewage or sewage treatment? Okay. Or you asked that question, right? The biofilm on sewage treatment. Did you get that answer yesterday? Okay, so Dr. He explained a lot of stuff, right? So it's the bioremediation function of biofilm, right? So the whole idea being is that biofilm, if you use the proper biofilm combination, it can trap a lot of heavy metal and toxic molecules, right? And that's how it can help. So that's a deliberate way of preparing biofilm so that you can remove toxic materials or toxic molecules from the sewage treatment. Okay. I use the arrow. I don't know if it's still not working to move it. Okay, is it working, or I can use mine? I have mine also. It's not working properly. Okay, let me. It's working now? Okay. I think other okay. That's good. Thank you. Okay, so I'll continue discussing the introductory part of the biofilm formation. Again, same. If you have any questions, feel free to ask. Okay, and make sure you understand what I'm going to discuss. So we talked about, how do I get out? How do I get out from? Okay. Okay, so when we talked about biofilm, we talked about beneficial applications, spoilage, and pathogenic application, right? Again, the way we are trained sometimes, we think the biofilms are bad, right? It's not always not true. So biofilms also can be used positively for your benefit. And I will also, hopefully during these 2 weeks of lectures, you'll find out, especially towards the end, we'll spend time on biofilm produced by probiotic organisms and how that benefits our health. So just let's overview a little bit of this different type of activities. Different type of, can you hear me now? So the sound feedback was strong. Beneficial and spoilage and pathogenic. So if you look in general, in the microbes, if you can group them, one we call it beneficial microbes, other we call it spoilage, and then the pathogenic microbes, right? And so if you look at, there's some overlap between beneficial and spoilage and the pathogenic organisms. I will explain that in a bit. So if you look at the beneficial microbes, it's pretty obvious to you, right? What are those organisms which we consider beneficial? Means it's good for your health or it has a positive attributes, right? So we're talking about lactobacillus, different species, then pediococcus, saccharomyces, and they're used for varieties of applications, right? And then if you look at the spoilage microbes, and their job is to spoil the food, right? And you can look at the typical spoilage organisms like Cyclobacillus. This is one of those organisms called spoilage of juice. Like if you like orange juice or apple juice, a lot of times many of the juice get spoiled by growth of those lactobacillus, and it's called Cyclobacillus. And this is highly heat resistant organism. And so in the food industry, this organism can survive in the processing plant. So when you are making large volume of juice, and sometimes they can be continuous or recurrent infection for the juice industry. And because they are highly heat resistant, it's very difficult to get rid of them. Okay, and moreover, they could form biofilms. It's one of the continuous source. And same way, you can look at the Brochathrix, Lactobacillus, Pseudomonas, yeast, and mold. So a lot of these organisms, we know they can cause spoilage. Okay, and we know how spoilage could be bad, right? I mean, you keep food in the refrigerator or on the bench, and after a few days, you'll find it that it's rotten, right? You cannot eat it. And food spoilage is going to be a huge issue going forward in the future, right? We all need food. For sustainability purposes, you need to minimize food spoilage, okay? So the other category is the pathogenic microbes. So if you look at all three categories, to me as a microbiologist, as a human, most important part is spoilage organisms. But it's most neglected, right? We don't study spoilage organisms, we don't worry about it, but in reality, as the world population continue to grow, right, the land is shrinking, the climate is changing, it's going to be unpredictable, and in order to produce food to feed everyone, we need to get all the resources possible, right? We cannot afford to lose our food. And if you look at the WHO statistics, sometimes they say 1/3 of the food we produce, they are all spoiled. Think about it. And even if you go to the grocery store or food processing plant, they also lose a lot of food. Individually at home also you lose a lot of food, right? I mean, sometimes you bought food and you forgot, or it's sitting there, it's gotten rotten, mold growing, or bacteria growing and rotting the food. So to me, as going forward, if you want to think about the future sustainability aspect of food or food security for the masses, we need to really think about food spoilage. How do we prevent food spoilage? Okay? Pathogens, we know is going to cause disease. We know they kill people, right? But many ways you can control it. Okay? Pathogenic organisms, yeah, I've been studying pathogenic microorganisms all my life. And I never thought that, well, that's a major problem to the humanity than the spoilage organisms. That's never true. So to me, I feel at this stage in my life, when I think about microbiology and future food security, food spoilage organism is going to be the key. Okay, keep that in mind. And so then the pathogenic, they are their own group. We know that the pathogenic organisms are food, and they come from varieties of sources, and they can cause disease. So one of the misperceptions is that if food is spoiled, let me just say this sharing of this, let me just point that out. What does this mean? This beneficial microbes and spoilage microbes share some characteristics or traits. Okay, let me give you an example. So the yogurt you eat, or if you had it this morning, and its primary organism is lactobacillus, right? And also Streptococcus thermophilus, that's two organisms. Also the Bulgaricus, sometimes people use that. And that organism is known for making yogurt, right? And you eat yogurt. Now let's see that. If that lactobacillus growing in a soup or some meat or some other products or cheese, right? Would you eat that food? You consider that as a spoiled food, right? Because that's unnatural for acidic, sour tasting piece of cheese or meat, okay? And so that same organism, when they grow on certain food, they can spoil the food, okay? And so that's why they share. So some beneficial organisms, we don't expect them to grow in certain food. If they grow, then they will spoil the food. So that's the sharing, okay? But they're never bad. But then let's look at the spoilage and pathogenic organisms. There is just slight sharing. So when the food is spoiled, the reason we don't eat it, one of the misperceptions we have, we say, oh, you're going to get sick, right? Are you following me? That's one of the reasons, right? Other reasons could be, yeah, it tastes bad, right? It smells bad. It's awful. You don't want to eat it. That's fine. But the point here is that sometimes people take this perception is that if I eat that spoiled meat or food or whatever food products you are looking at, oh, I may get sick. But that's not really true. Except because these pathogens, they don't spoil food. Next year you learn it today. Any pathogens we deal with, in general, most of the pathogens do not cause spoilage. So that brings us to a definition of spoilage. What is spoilage means, right? The spoilage organisms produce a lot of enzymes, a lot of other molecules, and that break down the food texture and structure, okay? and then it will produce some byproducts that become, offensive odor or bad smell, right? And that's the definition of spoilage. So if that's the definition of spoilage, do the pathogens cause food spoilage? So except for few, if you look at the sharing, for example, mold or fungi, they're number one spoilage organism. And the fungi also can produce mycotoxin. So they can spoil and they can produce mycotoxin. So that's one of the examples. There is another one is bacillus. Bacillus can spoil the food and also can produce toxin, can cause disease. Okay. There is another one which is called Clostridium. You heard about that, right? Clostridium. You heard about Clostridium botulinum? Clostridium botulinum produces botulinum toxin, and that toxin causes paralysis. So certain food, this organism would grow and produce toxin, and if you consume it, it's going to affect your nerve, and then you become paralyzed. And people can be paralyzed for life, or people can die. You heard about tetanus, right? Tetanus toxin, that's also produced by Clostridium tetani, that's the toxin. That also affects the nerve pulses. So, Clostridium botulinum, the reason I mentioned that, it also called proteolytic botulinum. That means some strains of Clostridium botulinum produce proteolytic enzyme. And like I said, the food protein or food products can be broken down by the protease. If the protease is present by this Clostridium species, then it can spoil the food. And at the same time, it can also produce toxin. But then there is a non-proteolytic Clostridium botulinum, which do not cause spoilage, but it can produce toxin. Did that give you some idea? Okay, so that's why the pathogens of their own. And generally, we get sick by eating certain food. That food is look apparently pretty good, right? Whether it's a salmonella diarrhea. If you knew there is a pathogen, you're not going to eat that food, are you? Like if you are buying street food or fuchka, right? Or street food, whatever you like, your best food, right? From the street. A lot of times the water, the type of ingredients they use, you are likely to get, Shigella or Campylobacter or sometimes Salmonella, right? And that food you eat it without knowing that there is a pathogen, right? And oftentimes you find out days later that you are sick, and likely I got it from there. But that food was not spoiled, was it? No. So that means pathogens don't spoil food. This is one of the misperceptions. I try to make it clear. For many people think that spoiled food make people sick. Is that true? Spoiled food, we don't eat the spoiled food, right? So there is no chance of getting sick. And first of all, the organism which cause spoilage, they don't make you sick. Okay? Keep that in mind. And that helps many ways, okay, in order to study these different microorganisms. What's their role? Okay? Let's use some examples. Well, I already talked to you about this. These are some beneficial microbes, right? And of course, you see the Swiss cheese, yogurt, and fermented beverages, pickles. These are all beneficial microorganisms. Again, I know I'm trying to think in context of biofilm, right? We'll talk about that in a minute. But a lot of this fermentation is not by single organism. A lot of times you have a multi-organisms, they're fermenting, they're producing this product. Okay, so beneficial microbes, they can also produce biofilm, okay? But those biofilm, we need them for the purpose of fermentation. So what they do, they can attach to the surface of the product and then form a nice biofilm. Immobilize, and then they could help in the fermentation process. Do you see this one? How biofilm formation can help make all those beneficial products. What about gut health? Right, you heard about gut health, right? Nowadays everybody is studying microbiome, microbial community, and how that affects your gut health. Because gut-brain axis is very important. Gut, skin axis, gut, every axis. Everything starts in the gut, right? So gut health can be improved by improving your biofilm formation in the gut. Okay, so we are talking about colonization. We are also talking about persistence of certain organisms in the form of biofilm in the gut. Everybody clear on that? So that biofilm formation is going to be very important for gut health. Skin health. Okay, what about skin? We know that, some people have very beautiful, nice skin. Some people suffer from skin infections, skin diseases, right? Varieties of diseases, right? And if you have a nice, healthy microbial community colonized on the skin, and generally that helps keep you healthy, right? Healthy skin. So, do you use soap all the time when you take shower? I know this is personal hygiene, right? So, depending on which weather are you, and you have to use soap, right? But do you know the soap is bad for your microbial community on the skin? They do. They're really bad. Because I know they're trying to sell you antibacterial soap, right? And you put a lot of soap on your skin. But in reality, that's going to damage or destroy your microbial community, which are the good one, on your skin. Okay? And I think there was a study, I think this story is very beautiful. This is in Brazil, and there are a lot of those community, they live in the forest. There is they don't have any kind of communication with our world. They live in a different world, right? And so there was an experiment. It happened to be the experiment done in a way which is kind of like unusual. So one American explorer went to that forest, okay? And he fell in love with the local women, okay? Local tribal women. And they got married, okay? And when they got married, and so they had a baby. And so, of course, he's a tribal plus American white mix, right? And then there was a time, so the lady was moved to US, okay? But it's a totally different world, right? I mean, you high building, totally different lifestyle, right? So one of these days, what they are doing actually, the son grew up, and he's now a student at Stanford, and he's actually studying medical, and he's doing microbial community study. So he helped this scientist, the team of scientists from US, to visit that rural area, okay? Because the language issue, they don't want the foreigners for some reason, and so they don't want them to study their skin microbiome, which is very important, that how these people, they never suffer from any kind of skin infection. They live in a very isolated rural community, and they have a beautiful microbial community. And so people are trying to study that microbial community, how keep people healthy looking skin. Because the skin and cosmetic industry is a billion-dollar industry, multi-billion. So everybody is trying to find a way how to maintain a healthy community on your skin so that you have a beautiful shiny skin. So the bottom line of the story is that this lady, she was brought to America for just 2 days or 3 days, and she's supposed to come to the lab, and they will take her skin sample. What she did actually in the meantime, as soon as she arrived to the hotel, and somebody gave her a good shower, with the soap and shampoo and everything. And they took sample from her before that, taking bath or shower with the shampoo and soap, and that community, and as soon as they took the sample right after shower, that community was shifted totally different. And that was like just one shower with this soap, okay? And that changed the community. So what I'm pointing out is that this sort of extra stuff we put it on, it really affects the microbial community. So the skin health, the community of the colonization of the native microorganism, which you perceived as a healthy one, they usually keep your skin healthy, okay? And what people are trying to do is that introduce that community or try to maintain that community on your skin so that you are bright, young looking skin. Right? Remember, as we age, our skin wrinkles, right? And we will try to have a shiny, beautiful skin, right? You heard about Botox? Botox, right? People take that for wrinkle, right? People getting old, they don't want to be old. But skin is the reflection of your age, right? If someone ages, the skin get bad. And if you want to keep your skin healthy, think about you have to manipulate microbial community so that they can keep it healthy. I know I told you a long story, but the bottom line here is that you have to pay attention to this microbial community and biofilm formation on skin in order to keep it healthy. So you have to understand that as well, right? And so that study, so I worked with several MDs, and the person who is trying to run this company, he's a medical doctor, and we had a long discussion. Then there is another medical doctor who were collaborating and discussing, and he said, "Oh, when I take shower after this talk, I don't use soap." Okay, that's the bottom line of the story. But again, if you are sweaty, you are doing athletics or you are running around, and yes, I understand that you need the soap, but you have to figure out what is the best way to use that soap so that I don't disturb my community. And what it does actually, it keeps your skin healthy. Some people looks, the dry skin and looks awful, but you want to keep it shiny and healthy. A lot of times the natural microbes, they do keep it healthy. Okay, you figure out how to do that, but I'll give you the clue. Okay, that's not the topic, right? Okay, skin health is very important. What about wound, right? Skin wound, right? Or the wound anywhere throughout the body, whether you accidentally cut it or there was infection. And can we use biofilm strategy to repair your wound? Okay, we have to understand that. So there are a number of ways you could use it. So I will show you some data that if you have a wound, create a wound, can you use certain probiotics to help repair your wound? It's amazing. Same thing that some of the cosmetics industry, I don't know, you can check it, those who like cosmetics, and there are some cosmetic industry, cosmetic products, skin health product, they are using probiotics, okay, live probiotics in that, and they are claiming that those probiotics can improve your skin health, biofilm formation, and then the better skin production and keep the diseases away. So these are some of those beautiful examples of biofilm as a beneficial. So let's talk a little bit about fermentation. I may spend that later on. So you know about fermentation, right? I mean, if you are making beer or wine, alcoholic beverages, right? Fermentation process, or a lot of byproducts, right? Fermentation is very important. So you might ask that question, how do biofilm help fermentation process? Okay, I think yesterday's lecture in the afternoon, he gave some clues, right? So one of the things is that if you add your microorganisms in this fermenter, and can you keep the organisms in places where it can help you convert the substrate into the byproduct you want, okay? And if these organisms are individual, they may not produce that type of effect. So it's designed to produce better byproduct for the fermentation purposes. Okay, that's an example. So this is what I was trying to tell you. This study is fairly new. You can see this 2024. So what they did actually, you can manipulate the organism. If you have, say, yeast you are using, right? And can you make this organism to form biofilm on the side of the vesicle, right? So this is a biofilm adapter. So they designed it in a way, make sure this yeast, they form biofilm, okay? But now I don't want the biofilm all the time, right? I have already got my product. Can I change it? Now you can break down that biofilm dispersal, and then you can manipulate another gene. So these are biotechnological development, just giving you an idea that we could use that for producing byproduct. Okay, so this is the beneficial application of biofilm. Okay, role of yeast in biofilm. So again, there are good thing and there are some bad thing, right? So here, this slide, I don't know whether you'll be able to read it from the distance. I know I just copied it from this paper, and it's saying the negative role. Yeah, if you have these organisms, and on wine, it can make off flavor, off odor, bad taste. A lot of things can happen. But also for positive role in wine making, yeast has a positive role. And so all of these different products, you will see sometimes biofilm works very well, it's desirable, you want it, right? But you need to know at the same time what negative aspect of that. So again, always scientists, if you're trying to develop a product, make sure you have a clear understanding. Make sense? Okay. Again, a lot of times I try to give you ideas, okay? So you can go and read and learn more in depth. So this is mixed organisms. We talked about polymicrobial biofilms, and these are some of the spoilage you will see this, right? These are again examples of spoilage. And then the pathogens, if I look at it from disease standpoint, and if you look at the diseases associated with bacterial biofilm, okay, and this is an incredible list, okay. You will see this atherosclerosis, right, heart problem, right, heart built up, infective endocarditis, then typhoid fever, all of this inflammatory bowel disease, colorectal cancer, prostatitis, wound infection, and you can see the ear infection, skin infection, urinary tract infection, mastitis, cystic fibrosis. So a lot of diseases are attributed to biofilm produced by pathogens. Okay, and so these are some of those lists I gave you. Here is that in animal system as well. Not only the humans, also the animals. So you are getting at ear infection, eye infection, urinary tract, respiratory tract, reproductive organ, all of those places, nervous system, all of those diseases can be caused by the biofilm forming pathogens. These are also wound infection. Wound is pretty common, right? We all suffer from. Animals also can suffer from wound. And these are also a good example of biofilm formation on the wound. Okay? Everybody clear? Okay. Now let's look at the little bit of the diseases. Do you have any questions so far? No? Okay. Let's talk about the diseases. Sometimes, of course, we worry about the food safety and a lot of the pathogens. And by now, you already know a lot of those pathogens, we call it zoonotic. And by definition, zoonotics are those organisms come from animals. Okay? And we are in contact with animals. Whether you are a vegetarian or non-vegetarian, we are always in contact with animals, right? So let's go over this. So these are meat animals, right? Meat, milk, egg. You can get these, right? Those who like, non-vegetarian, yeah, they can get a lot of these organisms through this. Well, these animals also carry these pathogens, and they can also contaminate soil and water, right? They are excreting these organisms all the time, and these organisms can go to plant and any of those plant-based products. Okay, so that means you don't have to be a non-veg to get the pathogens. You can get it through the fruits and vegetables as well. Okay, and that's a major problem nowadays. And of course, the companion animals, right? We have horse, cats, dogs, and we always keep them as a pet, and they also contribute to many diseases. Okay, and of course, the wildlife. We already experienced COVID, right? And COVID, we all recognize that it comes from bat. Bat is the primary source. Then there are many diseases like Ebola, and then also Nipah virus. I think India had few cases. I think last time when I was visiting, they were talking about Nipah virus outbreak in 2023 probably. There's a huge outbreak. And so those are, bat is the main source of Nipah virus because they are the one carrying it. Okay, and so wildlife, they become also major source of many of the pathogens we worry about. And so any of those organisms coming to us somewhere along the road, they are always going to have a biofilm. And as I mentioned before, always microorganisms form polymicrobial biofilm. They never exist in isolation. And I think this is just a broad scope. You heard about One Health, and I'm sure all of you are familiar with. So to me, I think One Health approach is to not one entity, one expertise can help resolve some of these problems. Okay. To me, I think if you ask a veterinarian, they worry about the diseases of those animals, animal health. Okay. They barely worry about humans. Okay, but they can be. But then a lot of those organisms comes through food products and then the plant-based materials, right? Plant-based foods. And so the horticulturists, agriculture scientists, they have to manage how this pathogen transmission can be blocked so that human can be protected, right? Same way the food scientists, food processing engineers, they try to find a way to kill all of these organisms. And then the medical doctor at the end, right? Do they understand the food system? How much do they know about food? How much do they know how these organisms transmitting through food to humans? Very little. They don't have much idea how these organisms transfer from one place to others, unless somebody is really reading up a lot of this stuff. So what I was trying to point out is that not a single entity can help resolve this major problem we are facing today. So to me, I think the approach would be is that all 3 or 4 different expertise needs to work together to help resolve this. Okay, medical doctor have no idea how these pathogens grow in the food and how food can influence the pathogen behavior. Right? We know what pathogens do to the food, right? If you have pathogens in the food, we just discussed, right? So they are there, but one aspect you need to start thinking, what does food do to the microbe? That's a big question. Always start thinking. Pathogens come through food, right? They want to eat food. You got that, what I'm trying to talk to you? What influence food has on microbes? Have you ever thought about it? It's like, let's put it this way. You know what influence your dad has on you. But do you know what influence you have on your dad? Very simple way. A lot of times you don't know, right? So same way, ask that question. If these pathogens come through, say you drank a glass of milk or ice cream, right? You took a bite of ice cream. And so ice cream may have some microorganisms. So if it is a spoilage microbe, yeah, it's going to cause spoilage. You know already it's bad, right? But if there is a pathogen, what effect ice cream has on those pathogens? Listeria is one of the organisms that comes through ice cream. Have you asked that question? So you are drinking a juice, bottle of juice, orange juice or apple juice. So if you have organism in that juice, right, likely say we had E. coli outbreak with the apple juice, orange juice also. People died or severe infection. What influence that orange juice has on that E. coli? Have you asked that question? If you haven't, if you are a microbiology student, you are thinking of microbiology, sometimes studying this, those are very important. I'm going to discuss a little bit about those, some of those. Okay? But that is the main problem nowadays we face. Why we suffer from certain diseases. Okay? Because you don't have an understanding of the disease. So this is like a, for example, this is also trying to tell you that pathogens in the food, right? It's a soft cheese. This is a salami or meat product. This is cantaloupe I was telling you yesterday, and the pathogens go from here. So one question always have to ask, and I think you need to start thinking seriously that if you have listeria or if you have salmonella or E. coli, any of those food, think about the composition of this food. What ingredient we have, how they are made, what is the pH, what is the salt, all of those components in this food. Do you think that's going to affect the pathogenicity of those organisms? Because you are all studying pathogenesis, right? And we are studying biofilm, and that's one of the big part of it. So if you have a juice, what is the pH of juice? Let's see if anybody knows. Acidic, right? Somebody said acidic. So that's pretty low, right? So give a range, close to 4, 4.5, 5. That's good. So that's a range, right? So that's acidic. Okay. What about salt? We have salt in certain foods, right? We add salt as a preservative, right? This is also, we can add salt. And then how else we save our food or store our food? A lot of times you do vacuum packaging. You heard about that? You remove the oxygen. Why do you do that? You remove oxygen so that oxygen-loving organisms, which are aerobic, they cannot use that, they won't grow, right? Okay, we are all on same page. Now, you drank that juice with E. coli in it. Okay, you ate that sausage which was vacuum packaged, no oxygen, right? And there was some bacteria there. What about our gastrointestinal system? How much do we know? We have acid in our stomach, right? And that we feel so comfortable that, oh, we have acidic stomach, and which is close to 1 or 2 pH, right? It's going to kill all the bacteria, all the microbes, right? And that's why I feel comfort. A lot of times I get these questions. People ask, oh, what about our stomach acid? It's low. It's going to kill all these organisms, right? Okay, what about our gastrointestinal tract? What about the oxygen level in our gut? So as you go down, a lot of you guys doing a lot of experiment in the lab, you know it's anaerobic condition, right? No oxygen, oxygen deficient environment. Now, if you have these organisms coming from anaerobic environment, coming from acidic environment, they are going to survive better in the gut, right? That's a very simple concept. So we needed to think what the food is doing to those microbes. So microbes are prepared well to survive better or overcome our gastrointestinal barriers. So they become acid resistant. They can be surviving without oxygen. They are anaerobic. So they are going to produce a lot of proteins as a stress response, and those proteins are going to help them survive better in the gut. That makes sense? And when you drink juice, you drink it, right? The bacteria don't stay there in the stomach much. Goes through, and they can cause disease. So these, a lot of these factors you need to pay attention to. And that's what I was telling you, that what food does to microbes. You need to start thinking. And that understanding going to tell you those factors can help also microbes to form biofilm. Because biofilm is a stress protection device. They want to protect themselves. So all of those environment I am creating tend to make these organisms robust for all environment they are going to encounter. Okay, so that's what I meant to tell you. And this is, again, this is the slides very small. I don't think you can read it. So what I was trying to tell you is that there's a lot of diseases caused by this foodborne pathogens. They cause varieties of diseases. You know, it's not just the diarrheal disease, which is very common, but if you look at the other forms of diseases like arthritis or hemorrhagic uremic syndrome or hepatitis, Guillain-Barré syndrome, this is called paralysis, flaccid paralysis, brain, encephalitis, meningitis, miscarriage, stillbirth, okay, pregnancy-related complication, neonatal infection, paralysis I mentioned, cancer, I mentioned that about mycotoxin, allergic responses. So a lot of those diseases, if you look at the list of these are actually food associated, foodborne. And even a lot of those, say Ebola, or COVID or Nipah, also we think they are also food related, because some of this food or drink we drink, and that's what the organisms are, and that's where we get it. And those are deadly diseases, and you hear about 70-75% mortality rate, not the COVID, the Ebola and Nipah virus. And so those are deadly pathogens, come through food and cause many diseases. Okay, I know I'm going pretty slow, right? So let's look at the chronic diseases. Dental caries, plaques, burnt skin, wound, heart valves, I mentioned that, cystic fibrosis, joint or bone replacement. And so all of those diseases, this is a pretty good example, dental problem, right? I mean, everybody experienced that. If you stop brushing for a day, you know that you get a tainted yellow, your dental built up. And you can clearly see that's nothing but biofilm, okay? And they formed a really strong biofilm. And if you clean it up, yes, this is look good. And then that's the dental, lot of dental problem. And dental hygiene is really important. I don't know how much you pay attention to. Again, this biofilm formation and a lot of those byproducts of this biofilm, they're in the mouth, right? And brain is sitting close to mouth. Did you notice that? There is a easy transit of a lot of those byproducts getting to the brain. And a lot of those diseases you don't pay attention to, but a lot of this starts in the mouth. Even for your personal hygiene, I always advise, mouth oral hygiene is so important for many of the diseases because of the close proximity of the brain to our mouth. And a lot of immune cells, and especially a lot of these byproducts produced by these carbohydrates from this biofilm, and they are absorbed. And they can activate immune system in a negative way. What it means is that they can cause septic shock, you heard about, and it's a high amount of a lot of those peptidoglycan, lipopolysaccharide, a lot of those molecules can be bloodborne and can cause severe problems. Okay, and so dental health is very important. Mastitis, you heard about mastitis? Inflammation of the udder. Okay, and mastitis also caused by the biofilm forming cells. And intestinal colonization, we talked about endocarditis. Enterococcus fecalis is one of the organisms they form biofilm. Cystic fibrosis is the lung infection, lung disease caused by Pseudomonas. And so this is mastitis. The milk, Staph aureus, or the large number of organisms, they actually reside in the teeth, and therefore they stay there. And when the milking happens, and then these organisms can end up being in the milk. Okay, and so that's the problem. And these are all due to formation of biofilm. Got it? And then also the cystic fibrosis, lung infection. Pseudomonas aeruginosa form strong biofilm in the lung. And those people who suffer from cystic fibrosis, and they definitely they have a problem with the breathing, right? The respiratory tract infection. And this is one of the major infections caused by Pseudomonas aeruginosa. Okay, so these are some of those prime examples of biofilm producing organisms, how they can cause varieties of diseases. Okay. Okay, so this is an example of catheter, right? And they're forming this nice fibrin-like structure. These are different microorganisms. This is cystic fibrosis. In this case, you can see these microorganisms and they're forming nice tight biofilm. This is in cystic fibrosis lungs by Pseudomonas aeruginosa. Okay. All right. Let's talk about how we're doing in the time. I forgot. It's supposed to go up to 11. That was the time. Okay. I might go a little longer. Okay. But we have a tea break, right? And then after tea break, I'm going to come back and I'm going to talk again. Okay. Okay. All right. Okay. Let's talk about the biofilm concerns in food industry. Okay. And obviously, many of you may have visited food industry or have some general ideas, right? How certain products are made. A lot of those, you can see the stainless steel setup, the bench everywhere. And if you don't clean it properly, everywhere the biofilm will be there. Like as soon as there is food, substrate, microbes find a room, find a place, they will grow, right? They will form biofilm. And so this is always a huge issue. And always you have to break down the biofilm and help, right? And so this table can help you understand a little bit. This is a study actually done by a medical professional, medical doctor, and they are looking for these pathogens on hospital environment. Okay, all of you can read this slide, right? I wanted to tell you that if you have a clean surface, okay, and if you look at that, Campylobacter can survive up to 6 days on a surface. Escherichia coli, up to 16 months. Listeria, several months. If you look at these, there are months some of these organisms can survive on the surfaces. What does it tell you? If you are preparing food or on the kitchen counter or in the food industry, anywhere, and you can see these organisms, whether they are forming biofilm or the spore, they can survive. And this is kind of fascinating. So that brings you understanding how the microbial behavior on different surfaces. You talked about persistence yesterday, but let's revisit it. So how do these organisms survive on this on the surface? So now let's talk about in a reverse order. So we're talking about like biofilm formation. Yes, they will form biofilm. They will stay there. It's difficult to clean them. Quorum sensing we talked about a little bit. We'll talk more about it later on. So they can communicate each other. They help each other, so they stay. Viable but non-culturable state. Yesterday during this talk, you heard about that, right? You all have some idea, viable but non-culturable. So if you work in a laboratory, you bring a sample and you plate it, and there is nothing growing. But if you change the conditions before you plate it, then you start to see they are forming colonies. So what it indicates that those organisms are viable but non-culturable in that standard process. You have to modify that. You have to know. So what we are finding out, this was first discovered with Vibrio. because they come from the ocean water, sea water, which is salty saline water. And these organisms convert into viable but non-culturable state. Now we are finding out a lot of those pathogens cause diseases, they also go through that state. So if they are viable but non-culturable, because you could not detect them, right? So that means you could not detect them doesn't mean that they are not there. But they are there, right? Everybody agree? So they are called persistent. So they are also forming a persistence in that environment. And this is, we have to understand more, why microbes do that. It's called stealth phase. What about stealth? Stealth means undetectable, right? And so their organisms go through that. And this is pretty clever. When you learn about that, it's so fascinating. What makes the organism go through that stealth phase so that you cannot detect them. And so they are part of this persistence mechanism. Spore formation, we all recognize that, right? Some organisms will form spore and they will survive. And stress response, I talked to you a little bit, but this is important. Stress response. So they can adaptation to pH, salt, acid, anaerobic conditions, antimicrobial agents, all of those, they can adapt to that. And that's also become part of the persistence. Are you getting it? So you add a lot of salt, you are expecting all the organisms are going to be dead, right? But not necessarily. Some of them can survive very well in the salt or in the acid, all of those conditions. And then of course the viruses, we kind of ignored it because they are not bacteria, right? But they have less complex structure, it's very difficult to kill them. If you have a complex structure, it's easy to kill, right? If you have a less complex structure, just the naked DNA or RNA, how do you destroy it? That's why viruses are very difficult to kill. Easy to kill bacteria than viruses. Did you know that? Viruses are small, but they are very difficult to inactivate because they don't have any complex structure. They don't have enzymes. Sometimes they borrow things from us. So that's why they are less complex, very difficult to inactivate. Okay. From the industry perspective, we're talking about how this biofilm. So this is a good example, right? If you look at the new hose, there is no built up. So this is from the dairy industry. And if you look inside the old hose, been used for many years, or many times, who knows, and you can see the cracks and then the built up. You don't see the inside the tube all the time, right? But they are forming biofilm. This is how they become persistent contaminant. This is some of those only stainless steel and this is also stainless steel surface. They form nice biofilm structures. These are also on stainless steel. You can see these varieties of population polymicrobial organisms growing. And this is on ice machine. I will end it after talking about ice machine. So do you like ice on your drink? You do? Now it is hard. One thing I do whenever I go to restaurant and they say they're bringing glass of water or drink and say with ice or without ice. I said without ice. I never take any ice. You know why? Because of this. Ice, we think that's really innocent, right? It's from the water, right? You never think ice could be a source of pathogens. We had outbreak with ice. And especially, even at your home refrigerator, they may have an ice machine in the back. And how often did you go and clean it? Never, right? Ever. Sometimes you clean it, yeah. If you're a good microbiologist, probably you go and clean it, right? How about in the restaurant? I mean, restaurant. Think about it. They're ever going to clean it? There's ice machine in the back, and first of all, you don't know what type of water they are using. Okay, that's the number one concern. The second concern is are they cleaning it? And if you look at this study, this just came out 2023. A laboratory ice machine has a cold oligorhombic artificial microbial niche for biodiscovery. And what this study is saying is that if you take different sample from different parts of the ice machine or ice, and you will find a lot of these microbes. And Pseudomonas is one of the notorious. Of course, the other organisms there, with their polymicrobial, and they're pretty sticky, and all those other pathogens can be trapped, including viruses get trapped in the ice. And so that's why always I hope I did not ruin your drink for today or next few days. Think about, oh, this is the ice machine. Dr. Punia said, be careful about ice. And it's true. Ice could be dangerous. Unless you really know, unless you have a refrigerator, you put a good bottled water and put it in a tray and made a ice, that's perfectly okay. But a lot of times these ice machines, they make and sell ice for drink or whatever purposes, they're not really safe. So what they did actually in this study, they did the microbial community study, and they're finding out at different temperature on that ice machine, because you have to convert it into ice, right? So you have to go to a 1 degree temperature, you find some organisms. 4 degree temperature zone, you find some organisms. Temperature at 15 to 25, you see a lot of these organisms. So what he's saying is that they're finding large community of organisms in the ice machine, which you supposed to consume all those microbes. So this is the problem with that, right? So anyway, I'll stop here. Then I'll talk a little bit about the stress. And enjoy your break. I'll be here. Any questions before you take a break? No question? Or you have some? You're kind of hesitating. Okay, if not, we can talk during the break and have your break, and then we'll start at